Well, I can still say good morning, everyone, and really appreciate the opportunity to talk to you today about my lab's work. Just going to give you a brief background for what my lab does and what our interests are and motivations. So I know this is a very diverse audience here today, so maybe we're not all very familiar, but the liver really is a very unique and special organ. When the parenchymal cell type, the hepatocyte, is injured either by drugs, toxins, viruses, or other insults, the parenchymal cell type, the hepatocytes, that are injured are replaced through the proliferation of undamaged hepatocytes. Now, when this process occurs either too acutely, it results in acute liver failure, or if it occurs chronically, it results in a process that we know as cirrhosis. Now, why this matters is 20 million Americans live with some form of liver disease. This number is actually higher in Europe. It's about 32 million Europeans. 3 million Americans live with cirrhosis. Right now, as of maybe about a few weeks ago, 10,000 Americans are listed for liver transplant, and 9,500 were transplanted last year. Now, I just want to point out this number gap doesn't look that big, but this is a huge jump in the number of transplants that we've traditionally done in the United States. The historical average is around 5,500 to 6,000. And what's accounted for this massive increase has been the opiate epidemic, which has been great for my patients, but obviously not a great solution for solving this problem. So as a consequence of this gap, still 31,000 Americans die each year from acute and chronic causes of liver disease. And worldwide, 1 million people die yearly from cirrhosis. So if you could imagine a world where we had an unlimited supply of human liver tissue or human cells, this could be used for a variety of different applications, such as that in the basic science or clinical practice arena. More specifically, we could use this potentially for hepatocyte or liver transplantation or for liver disease modeling. In both these contexts, though, insights from the basic science arena could feed into the clinical arena and vice versa. And this is all with the goal of trying to improve the quality of care and outcomes for our patients. Now, when you go about trying to think about how you want to build a human liver, and this is true generally for almost any tissue engineered organ, really what it comes down to when you boil it down to its basics are the cells that you're going to use in that system and the microenvironment that those cells are in. And if you drill down a little more deeply, it raises questions first where you're going to source these cells, how you're going to maintain their viability, stability, phenotype, and function, what non-parenchymal cells, in this case, what non-hepatocyte cell types you're going to include. And then it next raises the microenvironment that those cells are going to be in. How are you going to organize those cells? What growth factors are going to be built into that environment? What cellular or extracellular matrix components will be included. This is all going to impact how the cells interact with each other, how cells interact with their environment. And then it's also then ultimately going to dictate the mechanical properties of the system. And this is all before we start getting to questions about how we're going to enable nutrient delivery, waste removal, oxygen, things along those lines. And all these decisions are ultimately going to impact the global phenotype and the function of the mini tissue that we generate. Now, a lot of this conversation was moot before the discovery of human embryonic stem cells by Jamie Thompson's group in 1998, and then the development of induced pluripotent stem cells in mouse in 2006, and then in human in 2007 by Shinya Yamanaka. So this then offered us the opportunity to have a cell type that had the potential to become any cell type. In our case, we were interested in trying to make hepatocytes from this system. And the thing is that initially when I started working on this almost 23 years ago, I figured this would be a pretty easy problem to solve. And it literally took us in the community over 20 years to solve this problem. We kind of mimicked what was occurring in development. And by modeling how the fertilized egg becomes a hepatocyte or goes through each of the, breaks the germ layers and then goes from there, by modeling this, we can actually take a pluripotent stem cell and then make hepatocyte-like cells with high efficiency. And what we can show you here, or what I can show you here, is that we can take, as it goes through, it forms hepatoblasts that form islands of hepatocytes. Here you can see biocunicular networks that are forming the formation, and here blue is pointing out binucleated cells. And then if you follow this process, you can see as we go stepwise from a pluripotent stem cell, the iPS cell and then march forward through endoderm, hepatic specified endoderm, hepatoblast, and hepatocyte-like cells, we actually upregulate or lose the markers that are appropriate at each stage of the differentiation. Now, it's important that these cells just don't look like hepatocytes, but that they function like hepatocytes as well. And so what we've gone on to show is that we can actually produce cells that actually have robust hepatocellular function. This is on the protein secretion side, such as for albumin and alpha-1 antitrypsin secretion. But the one thing I want to point out, and I kind of summarizing like 20 years of data in like just a few slides, but one of the conclusions that we've come to is if you actually compare these to adult human hepatocytes and to fetal human hepatocytes, what they most closely phenocopy is that of fetal human hepatocytes and not adult. This is just giving an example here. If you do a bulk RNA-seq comparing primary human hepatocytes, fetal human hepatocytes, and the iPS-derived hepatocyte-like cells, that they actually cluster most closely with that of the fetal human cells. Now, if I look at this at a different way at the functional end, these are cytochrome P450 assays, both BFC and MFC, that encompass many different cytochrome P450 components. You can see that the iPS-derived hepatocyte-like cells do quite well as compared to a very robust primary human hepatocyte system. But if you start looking at like CYP1A, 2A6, CYP2D6, 2A6, you can see that they progressively get less differentiated function. I'm not even showing you CYP3A4 because it's just embarrassingly poor. So, if I take this, and I've done, metabolic functional analysis and all these other analyses, to cut to the end, what we're producing are fetal hepatocyte-like cells. And I just don't want you to walk away from this talk thinking that, oh, this group doesn't know how to make robust cells for the liver space. First of all, this is a problem in the stem cell community at large. If you go to a talk and someone says, I make adult cells, they probably aren't being completely honest about what they're generating. Now, some of this isn't necessarily our fault. We actually don't know how fetal cells become adult cells. So developmentally, we can't model a system that we actually don't have data for. And for that, I'll blame the developmental biologists, but more seriously, we just don't know how to do this. The other thing is just kind of the system we have. I'm expecting in 20, 30 days to phenocopy 10 months of human development, and that's just not how it works. More importantly, in the case of the liver continues to develop over the first 6 months of life. And so we also don't really know what's going on in that system. And so to generate cells that are more mature, I'm going to skip over this aspect, but obviously we're missing a whole variety of other cell types that are present in the system. We're also missing small molecules that are present in the system. We're missing extracellular matrix components that are present in the system. This is generally done in 2D and not 3D. So you take this all together, there are some limitations with the cells that we have. But what I hope and I've been able to show you is we can generate cells that have at least the phenotype of fetal cells. And the question then is how to build in these other components. So one of the first things that we've done is take the 2D system and then try to generate this more into a 3-dimensional system. And so we can engineer these plates where we essentially generate spheroids, or the community loves using the word organoids, but in any case, we'll just call them spheroids. And essentially, these spheroids, we can build in whatever different cellular complexity that we want into them. They're very uniform at the end of this. And this has allowed us to actually go for different stages of the differentiation process from the 2D system into a 3D system. And what we've seen is that we can actually promote the maturation of the cells with loss of fetal markers and upregulation of a variety of adult markers. And the thing is, though, there's a lot of questions that remain open here then. All I'm doing is putting the cells together in this 3D environment. What about the other components that are now missing in the system, such as cellular organization or others, and other cell types that might be missing? And, for this, I have to say, what we did is we used the same organoid or spheroid generating system, but where we have the hepatocytes here in the center, and then around this we build the endothelium, let's say in this example, around this. And the question we're trying to answer is how do we organize these cells in relation to each other that promotes the best functionality of the hepatocyte-like cells or hepatocytes? we could have a system where it's 100 microns apart, or 3 microns apart, or one where it's just part of the spheroid itself. And I have to say, in this case, nature was good to us because the actual system that worked the best was when it was actually part of the spheroid itself, not one that was exactly 2 or 4 or 8 microns away, which would have made this a much more challenging engineering problem every time. So basically, so that was the first question we wanted to answer. So, okay, it can be part of the spheroid. Then the next question is, well, what type of endothelial cells should we build into this system? And I just want to say that this is all primary human hepatocytes and primary human cells here, such as using liver sinusoidal endothelial cells or just generic human umbilical vein endothelial cells, or in this case, just kind of the IPS-derived endothelial cells. And we found is actually that, maybe not unsurprisingly, that having a system where the liver sinusoidal endothelial cells or the liver-specified endothelial cells was the one that best maintained the differentiated function of the hepatocytes, in addition to that of the IPS, while the kind of the generic kind of workhorse endothelium that a lot of people like to use, the HUVEC, just were less supportive of primary human hepatocyte function. We next wanted to build in the Kupffer cells, which are the tissue-resident macrophages of the liver, into the system. Now, one of the challenges there is maintaining their viability over time, just even a simple question like this. So if we label the cells, we can maintain their presence over a 2 to 3 week period. But the question then was, first, are we still maintaining the hepatocyte function if they're present? And the answer is that we really don't seem to have any negative impact on the differentiated function of the primary human hepatocytes. But then the next question is, are the Kupffer cells that we have then in the system after a few weeks still behaving and functioning as Kupffer cells? And so what we did is we actually challenged these with LPS and then look for production of TNF alpha, IL-6, and IL-10. And you can see here that in the multicellular spheroids that actually have Kupffer cells, we get a very robust production of these 3 different cytokines. And that's all in the ELISA, so it's all protein. Now, again, our goal is to try to build complementary IPS-based systems in addition to these primary-based systems. So we wanted to see, could we do the same thing all generated from IPS? And so using pluripotent stem cells going through this protocol, we can actually sort out cells that are CD14 positive, CD163 positive, CX3CR1 positive, and CD11B positive. And when we sort these cells out, they have the FACS, phenotype of macrophages. But if you actually look, if we pull out the monocyte compartment macrophage or purified, you can see that they have the appearance of macrophages, but importantly, they function like them as well. If we actually challenge them with LPS, they will produce the appropriate cytokines. But if we actually also add LPS and add, basically FITC-dextran beads, they actually will phagocytose these as macrophages should. We can do the same thing with endothelial cells. And essentially now we can generate, these multicellular spheroids that, have, Kupffer cells long-term. This is just an IF image of that. And, they don't adversely, first of all, impact the function of the hepatocytes longitudinally over a 1-month period. And we can also do the same from the IPS system. It's a little less functionality as what we have from the primary cells, but we get similar results. I think the most interesting thing, though, with the IPS-based system is the added functionality that we get as the cells sit together and interact with each other over time. This is a very complex, this one thing, so I'll go over it in a second. But if we just add LPS to the system, the spheroids that have the Kupffer-like cells or the macrophages respond appropriately, just as I had shown you earlier. But what was interesting is if I look on the hepatocyte side, the multicellular spheroids that have hepatocytes produce factor 7, which is a coagulation factor produced by hepatocytes. And I can antagonize that with a vitamin K antagonist, so it's appropriately produced and responsive. Now, what's interesting is the multicellular spheroids that have the endothelial compartment built in actually produce factor 8. Factor 8 is produced by LSAC in the system. The thing that was interesting to us, and we're still trying to figure out what this crosstalk, is that IPS-derived endothelium don't produce factor 8. So it's only after 3 weeks of culture with the hepatocytes that we actually ultimately see the production of factor 8. And so when we look at those endothelial cells, they're becoming more LSAC-like, But what the crosstalk is that's driving that, I don't know. So we're still trying to figure this out molecularly. But what I think, at least I hope I've been able to show you, is we can build these multicellular systems that we can maintain their viability and function and phenotype over, a multi-week period. And basically, as you all are aware, traditional culture platforms generally don't support hepatocellular viability or function long term, nor the non-parenchymal cell types. And moving into the future, what we've been trying to do is actually build these microfluidic-based platforms. So this is actually a vascular network here where we actually have flowed in V cadherin on both sides, and it's labeling this from inside the perfused capillaries essentially that are formed. And we can actually incorporate stromal cells into this, which is what the purple is here. And now we have now been slowly building in more and more hepatocytes here, which are shown here in green. So it's kind of just how we've been trying to push this platform forward. Now, there's obviously a lot of different applications I can talk to you about today, and I'd be glad to talk to you about it offline. I'm just going to talk to you today briefly just about how we've used this to study infectious disease. So one of the, about half my lab works in infectious disease questions. And one of the viruses we've spent a lot of energy on over the last decade focusing on is on Hep B. Now, not everyone here might know or be aware about Hep B, so I'm just going to take like less than a minute to discuss this. So Hep B is the most common viral hepatitis globally. 2 billion people have been infected with Hep B worldwide. About 250 million have a chronic infection. Despite there being a vaccine, there's still 4 million new cases per year. There's 1 million deaths directly attributable to Hep B. Hep B is the 10th leading cause of death globally. And the thing is that clinical therapy is targeted towards viral suppression, not cure. So basically, if we have to treat people like I do, it's generally for life. And the therapies are, thankfully not very symptom-free, but they're not trivial. They have to be taken reliably or can cause pretty significant impacts. Now, what's been the problem with studying Hep B has been that the natural host for the virus is the human liver. But what the community has used for the last 40 years to study this virus is this cell line, either HepG2, which is a human hepatoma cell line, or Hu7.5s, or the HepG2-215 cell line. And as you are all aware, these cell lines are marked by abnormal proliferation, unpolarized cells, dysregulated gene expression, and aberrant cell responses. And the impact of this has been that we really haven't been able to study how the virus, enters into the cell, various components of the life cycle, hepatocyte, HBV, crosstalk, et cetera. And I mean, the impact has this been literally a virus that we've known about for 50 years. We did not actually know how it got into the hepatocyte till 2012. So, I've basically been really focused on the fact that I really do think we can do better using the different platforms that we have in the lab, and we wanted to build a model system that could recapitulate all aspects of the HBV life cycle that would enable us to study the crosstalk between the hepatocytes and HBV. And so, I mean, just so you know, everything I'm showing you is going to be with IPS, but we've done the exact same stuff with primary human hepatocytes as well, and that part has been published. So I can talk about that if there was interest. But I've already shown you how we can make hepatocyte-like cells. But what I wanted to say is the entry receptor, which, as I mentioned, was discovered in 2012, is a bile acid reuptake transporter called NTCP. And it's expressed later on in the differentiation process. And importantly, in our multicellular platform, for both the IPS or the primary human hepatocytes, expression of this entry receptor is maintained, while in traditional hepatocellular culture, it's lost usually within 3 days. So importantly, if we infect our different cell types here as they march forward in the differentiation, it's only after they acquire NTCP and later on in the differentiation process where we can actually produce the HBV RNA and HBV protein. And I realize I took out the slide with the life cycle. So basically the virus gets into the cell, it gets to the nucleus, that the DNA that the virus gets repaired, produces this plasmid-like structure that ultimately it's called cccDNA. This leads to the production of RNA, which is there's 5 different RNAs that are produced. The longest one is called the pgRNA, or the 3.5 kb RNA, that's used to make protein, or it can be brought into the virion to be made into a DNA through a reverse transcription process, package, and then you produce a virion. So what I'm showing you here is that you're producing both the viral protein and that viral RNA. And the field's a little old school, so we still do southern blots for the HBV DNA. And you can see that there's a very thin band there. I have better on the next slide. And you can see the protein staining here. Again, field's a little old school, so I'm showing you northern blot, southern blots here, and basically showing that we actually can produce each component of the life cycle in the system. And then in using traditional culture systems, you just cannot actually generate any of these components. Now, the one powerful thing with the IPS platform that is something you can't really as easily do with the primary human platforms is leveraging the power of CRISPR to actually engineer the different, mutations or variants that you might be interested in studying. And so I've shown you I can make hepatocyte-like cells from this. What we were interested in was trying to understand the different polymorphisms that we're able to manipulate or alter viral entry. And so basically we generated IPS lines that had deletions or essentially mutations for ApoH, ASGPR1, and NTCP, basically just showing you on the RNA level that we can actually eliminate this for each one. And I'm showing you here the Western here for showing for specifically the NTCP line. And so we basically assayed now, we took these lines, made hepatocytes from them for each of the deleting lines. And what I can show you here is that for the NTCP knockout line, you just lose all ability to infect these hepatocyte-like cells, which is different from the ApoH and the ASGPR1 line. Now, if I add entecavir, which is a inhibitor to reverse transcription, it blocks the virus from replicating, we can actually block these other components and actually eliminate infection in the ones that were previously permissive, showing that this is real. And again, only in the NTCP or the entry receptor knocked out line can we actually do this. Basically, if I then want to engineer into the iPS cells different SNP variants that are seen in the human population, we can do that as well. And so there was a paper published in Hepatology that showed that there was this variant in the entry receptor, NTCP, also known as SLC10A1. And it basically provided resistance to chronic HBV infection. And there was another paper that talked about this isoleucine threonine line that could do the same. Although that was at one single patient, while the previous paper was about 15 patients. So we actually engineered lines with these different mutations. And what we found is that the serine phenylalanine mutant line actually also had inhibition to infection, while that isoleucine threonine line, which was just a single patient, probably something else is going on because we could not replicate that, at least in vitro. And basically, basically what I just wanted to show you here is that using either primary human hepatocyte platforms or the iPS-derived hepatocyte-like cell platforms can actually allow us to generate productive infection. And that, NTCP expression, again, is important in this process. It's really more just to show you this platform as a tool. I'm going to, just because we started like a few minutes late, I'm going to skip over this aspect. The last thing I just wanted to show you here is this slide. So using that multicellular platform I had shown you earlier, where you have just hepatocytes with or without Kupffer cells, we actually generated these same infections. And we actually got very surprising results, which we are currently working on right now, which is in the primary human hepatocytes, we get very robust infection. But if we add Kupffer cells to the system, my hypothesis and what we thought was going to happen is, well, here you have this, immune cell that we're putting in, this tissue resident cell, and we would actually have an impact on the infection in a negative way. And we actually saw the exact opposite. It actually enhanced the infection in the system quite significantly. And basically, this has been quite reproducible across multiple different donors. And by the way, this is all primary, so this is not IPS-based. So basically, this has been true, and we found this across 5 different donors. And just so you don't think this is some weirdness with just Hep B, we have similar results with Hepatitis C and other hepatotropic pathogens. And so basically, when we tried to dissect this in a little more detail, we found that there was a massive increase of IL-6 production by the Kupffer cells during HBV infection. And if we actually used a neutralizing antibody to IL-6 in the context of these infections, we actually like lost that increase in the amount of infection. So somehow this crosstalk in IL-6 production is leading to this process, but the mechanism we're still trying to delineate. So, with that said, I really do believe that these systems can really help, elucidate really novel biologic questions. Both the primary systems and the IPS systems, they both have their advantages and disadvantages. And, we've been using this to try to tease out questions as it relates to molecular virology questions, as well as questions as relates to metabolism and drug injury, which is things I haven't spoken about. Basically, it's taken an immense amount of people to kind of do different aspects of the work that I've shown you today. So I just want to acknowledge my collaborators in this and my funding sources. And with that, I would gladly take any questions.
Thank you, Robert. That was a great presentation. We do have time for questions, and I have a quick one myself. And maybe I missed it, but what about using L-sex, primary human L-sex with the stem cells? Does that make them more mature?
Yeah, so if you're saying taking iPS-derived hepatocytes and adding human L-sex,
Yeah.
so we've done that experiment many times. For whatever reason, that does not work out very well. The iPS-derived hepatocyte-like cells don't like that environment, just being alone with the adult cells. I think that the reason why it's worked with the IPS is they're also immature.
Mm-hmm.
So putting an immature cell with another immature cell has worked out pretty well, but mixing the adult LSEC with the IPS derived system, at least we haven't figured out how to get that to work as robustly as we'd like.
Interesting. Any questions? Back here.
Thank you. Have you ever tried stellate cells in your model?
Yeah, so I didn't show that data today because that would be like a whole hour presentation, but we have added stellate cells back to our system. I mean, one of the problems with stellate cells is I kind of view it as Schrodinger cell almost. If you have a stellate cell, it's probably not a stellate cell anymore. It's probably been activated somewhat into a myofibroblast. And so we've put a lot of energy into trying to figure out how to create an environment that allows it to reacquire some of its quiescence and then put that in the system. Because if we put like an activated myofibroblast into our system, what we're really looking at is not a normal system, but more of a, activated, more of a fibrotic system. Yeah, so basically, I mean, so we use IPS-derived stellate cells that have been made through the protocol of Pau-Sancho-Bru. And I mean, we can generate these systems and we can ask questions related to fibrosis activation and things along those lines. And I mean, it does work well. It just what I would like to have, and maybe it's just, beggars can't be choosers. Like I'd like to have a system that is as normal as we can have, and one that then we kind of are playing around with the system. And right now, the way I see it is we have systems where we're kind of somewhere in the middle, which is fine for the questions that we're trying to address. So I agree with you.
So interesting talk. So the mutation in this patient, the Berlin patient in AIDS, basically this is similar to this one. So did you say you've been able to reproduce it in the IPS or not reproduce it?
Yeah. So the phenylalanine. So there were two mutations we were examining. I know I breezed through that pretty quickly. So there was a here I can go back to it real fast, but So the serine phenylalanine mutation, we were able to reproduce that phenotype, and but the isoleucine threonine mutation did not reproduce what was reported in that paper. But the thing is, I think it was an N of 1 patient, and they were attributing the resistance in that patient to the mutation, and I think that might have been an error. But the serine phenylalanine, there's like multiple families with this, and they all had resistance, and so we were able to reproduce that phenomena.
Have you been able to isolate adult hepatocytes from people and look at these mutations or something, or this is in the plan?
So, I mean, some of these mutations are pretty rare. So like the serine phenylalanine, we definitely could. The isoleucine threonine, there's been a single patient that's ever been identified with that. So I think it'll be harder to reproduce that specifically, but the serine phenylalanine it's like present in like 1 in like 0.01% of the population, so it'd be possible. We haven't done that, but.
Any other questions? We have time for one more.
Yeah. Quick question, follow up on the issue of the integration of adult LSACs. How about attempts to transplant these organoids into liver? How have they worked?
Yeah, so basically the full adult system works great. We can get very good integration and repopulation and engraftment in a mouse, in a mouse model that would support that type of integration. Basically the iPS-derived hepatocyte-like cells, on the other hand, that's been far more challenging regardless of the different systems that we use. So if we do it in an orthotopic type setting, we can get engraftment and survival of our grafts. But the thing that we want to see is, let's say, maturation and growth. And that's not something that we've been able to do reliably. And, that's just not just us, that's the community at large. So if someone here has solved that, I would love to talk with them, because, it's something that we in the community has really struggled with.
Thank you very much, Professor Schwartz.